Stem cell. What is a stem cell? Well, stem cell is a very original cell which can make every type of a cell in our body. Stem cell also has the ability which can replicate forever, so renewal property it contains together. When did we start to use stem cells? Well, actually it has quite a long history, almost 100 years of history we have for bone marrow transplantations. If you look at the bone marrow transplantation steps, in the beginning, we collect the bone marrow from the center of the bones. And those bone marrow material is injected into recipient. Then those bone marrow moved to the spleen, and eventually they make all kinds of blood cells in the recipient. Next step, if you take those cells from spleen and inject another recipient, now the second recipient also develop all kinds of blood cells in their body. While the bone marrow contains the stem cell which can make a different type of blood cells, the very original the stem cell, the embryonic stem cell, is made from embryo. When the egg cell and sperm cell fuse, the fertilization happens, then they develop into blastocyst stages. In the blastocyst, the inner cell mass can be collected, and these cells are the source of embryonic stem cells. Embryonic stem cells, they can make, in principle, every cell type in our body. If you think about the potency of a stem cell, the fertilized egg cells, they can divide evenly 2, 4, 16, 32. Up to this stage, we call them morula stage. And then they started the form of the blastocyst, and the inner cell mass make embryonic stem cell, and embryonic stem cell can make every type of organs and cell types in our body. That's why this inner cell mass, those embryonic stem cell, is a pluripotent cells we call. But interestingly, this inner cell mass itself cannot make the baby. So to make the baby, actually it needs a little more cells, and that's why this blastocyst cell is complete potency it has, that is called as a totipotent cells. Now, if you look at the stem cell potency for this blastocyst, now we can say the inner cell mass is provided the embryonic stem cell, but also it requires trophoblast cells outside of this, the inner cell mass area. Then we can figure out inner cell mass make the embryo structure, but trophoblast make a placenta. So with this placenta, now this blastocyst has a totipotency and it can make embryo. It's very interesting to see here. Placenta is not the mother's organ. This is originated from embryos. Quite interesting, isn't it? If you summarize, the fertilized egg cell passes the totipotent stage, and inner cell mass can be isolated to be cultured to make embryonic stem cell, which is pluripotent. And these pluripotent stem cells, they can be divided into ectoderm, mesoderm, endoderm, 3 layers. And endoderm eventually can make all the internal organs. Mesoderm can make mesenchymal stem cell, also hematopoietic stem cells. Ectoderm make the skin and neuronal stem cells. And neuronal stem cell make all different kind of neurons. Mesenchymal stem cells, lipid cell or muscle cell. And this intermediate stage of stem cells, in another term we call adult stem cell, those can make some numbers of different cell type, but not whole cells. So this cell stage we call multipotent cells. Some cells, for example, satellite cell make only myocyte, and germline stem cells, they can make only sperm or oocyte. So if the lineage of one cell fate is fixed, then this kind of cells, the potency we call unipotent cells. To check the property of embryonic stem cell, we can do different experiments with animal cells. For example, we can check teratoma formation to confirm those cells has embryonic pluripotency. Also, the cells should have ability to make all three layers of the cells, ectoderm, endoderm, and mesoderm. Also, the cells, when it's mixed together with the other embryo, it should form chimera. And eventually, the cells should be transferred to the germline formations. That's a little more difficult, but this is the evidence of a pluripotency. Most difficult experiment is a tetraploid complementation test. If you pass this test, then it is confirmed as embryonic stem cell. Using human ES cells, it's difficult to put them into the human body, but at least these cells can be tested in animal body to check the teratoma formation or three embryonic germ layer formations. Now, chimera formation or germline transfer or tetraploid complementation is technically maybe possible, but it's difficult to perform for ethical issues. Let's look at the tetraploid complementation experimental details. Embryo grows through blastocyst, then eventually it can make embryo and placenta. In tetraploid experiment, we can give a shock, electric shock to the embryo. Then the cell division is not completed. So usually cells should have a 2N DNA diploid state. But if the cell division is not completed, then some cells may contain 4N stage of DNA. That's what we call tetraploid cells. Then inner cell mass cannot grow smoothly, so they die out. But trophoblast cells, they can survive. So here we can inject external embryonic stem cell and check whether the placenta is formed from original cell, but embryo is formed from external cells. If it passes this test, then it confirms that this external embryonic stem cell has pluripotency. So itself does not have the totipotency, but with the help of placenta by other the body, then it can form the embryo eventually. The major component of a bone marrow, they contains hematopoietic stem cells. Hematopoietic stem cells, it has a power to make all kinds of blood cells in our body. That's why if you have the leukemia case, then we remove all the patient's leukemia and then transplant the healthy person's hematopoietic stem cell using bone marrow, then it can rebuild new blood systems. Another popular source of stem cell is mesenchymal stem cells. Mesenchymal stem cell can make many different types of cells in the body, including muscle and fat and osteoblast. The ideal case is maybe utilizing embryonic stem cell for transplantation. So if we have an injury or accident, we lost some part of our body, then embryonic stem cell transplantation, in principle, it can make out any cells, any organs. But it has some potential problems. Embryonic stem cell is possible to make a teratoma. So you don't have liver outside of the skin or ear on the leg kind of things. Also, embryonic stem cell has potential immune rejection response. So if you use somebody else's embryonic stem cell, put them in my body, then my body may sense those external embryonic stem cell as foreign body, then it can induce immune response. To overcome the immune response problem, we can do maybe clone my own cells embryonic stem cell. How can you do it? Well, one possible way is we can collect our cells, our somatic cells in nucleus, and transplant that into empty egg cell. Then it can mimic the embryo formation that we can collect those embryonic stem cell and use. Another possibility may be utilizing somatic cells, kind of old cell, by giving some kind of a signal or stimulus, and we change that into artificial embryonic stem cell. This type of cell we call IPS, induced pluripotent stem cells. Let's look at nuclear transfer. Well, the pioneer of this work is John Gurdon. He utilized the frog cells, and then frog skin cell nucleus is injected into the frog egg cell, and that could form the whole body. So this kind of the transfer of a nucleus can transfer the genetic information into empty egg cells. For a long time, this kind of a nuclear transfer is believed to be only possible in reptiles or the amphibians, but it has been demonstrated utilizing sheep. So the first case is a dolly. So in dolly's case, they use the three mothers. The first mother is an egg cell donor, but nucleus is removed. Second donor is a nucleus donor from the memory the gland cells. Nucleus is isolated, and these two the component is combined to make a new fused cell. And they utilized the third surrogate mother, and then Dolly was born. Now, can you guess this Dolly has the same genetic information with which mother? Yeah, that is nucleus donors, the mother.